Alright, this week we are looking at roots and shoots and the sort of tissue organization and developmental pathways. Normally these lectures have tons of drawings and we draw all of the anatomy. With my point and click mouse situation here, I'm not really able to do that, so I'm going to show you pictures of the anatomy and talk about the differences, but I'm not going to do the sort of cartoon diagrams of the anatomy. So I apologize for that. I also apologize if there's any weird glitchy stuff. I was just on another slide, but my computer's being very strange today. So a little overview. We'll look at different types of growth, referring to primary and secondary growth, which are terms that may have come up in your labs already or in your reading, because the content is all connected. Then we'll look at the root apical meristem, which is right here on this growing root tip. Then we'll look at, we'll do some drawings. You'll maybe do some drawings, or you'll just look at some cross sections. We'll look at a root tip long section, so this way, right now. That's what we're looking at here on the right side of the screen. We will look at a root tip cross section, so going through it. We'll look specifically at the vascular cylinder, so the organization of the vascular tissue within the root. And then we'll look at a root in secondary growth, which you'll find out what that is soon. The last thing we'll look at is a flow chart of tissue development. Now normally I make students memorize this, and then I ask them questions about specific cells and I have them trace the order of tissues of development, but normally we focus more on that in a lab setting, so I'm not really sure how much this is going to come into play for our class. If you go on to be a botany major and go into like developmental plant biology, this is all content you would need to know, so I don't want to short shrift anybody headed that pathway. Types of growth. The primary growth happens in all plants. It's the main initial type of growth. Any plants that are herbaceous, that don't have woody tissues, this is the only type of growth that they'll do. So it happens in all plants. It results in an increase in organ length. So plant organs can be things like roots, stems, leaves. Those are all organs for plants. So a leaf could get longer, a stem can get longer, roots can get longer. There's not that much organ thickening. There is some organ thickening, like we know that plants do get a little bit wider as they're growing, but there's not like a focus on lateral expansion. And this is all going to be done with primary tissues. Secondary growth, on the other hand, happens in woody plants, so all gymnosperms and most angiosperms are going to, not most angiosperms, some angiosperms, a lot of angiosperms, but there's just a lot of angiosperms in general, are going to go through secondary growth. This results in an increase in organ thickness, so wider trunks, wider roots. Secondary growth does not happen in leaves, so those are excluded. We're just talking about roots and stems for secondary growth. And the last part, this involves secondary tissues. So primary tissues might give rise to some secondary tissues if plants are going to undergo secondary growth. But some plants will just do primary growth. Like we talk about monocots as being plants that don't undergo secondary growth. But some do make some weird woody tissues like bamboo. Okay, so the root apical meristem, the RAM, this is where new cells are produced through mitosis. Cells can divide in two directions. So for all of our meristems that we talk about, we'll usually talk about, there's going to be a caveat here, many of our meristems will talk about cells dividing in two different directions. When we talk about the outer, like, meristem that makes the epidermis, that one is maybe just one. Okay, so anyway, the root apical meristem. To the exterior of the plant. So in this case, we'd be talking about downward. This is where the plant will be producing more cells downward, and those would be the root cap. Sorry. So the you can see my mouse moving here. It should be a yellow circle. This sort of ring of cells right here is going to be our root apical meristem, and that is going to produce, as it divides, cells up and down. The downward produced cells are going to end up outside of the root, and they're going to form a structure called the root cap. And that's this little conical hat here sitting on the outside of this root. You can see it also here as these densely packed small cells that are sort of encasing the outside of the root. And it ends up running up the side of this growing root tip. This is a place where there's a lot of interaction with the soil environment. So the root cap protects this growing root tip as it's pushing down through the soil. There could be rocks or sharp things, tiny pieces of silica that could damage a growing root tip. So having this kind of constantly produced mucilaginous sheath of cells on the outside of the root helps pave its way. So it gives it some protection from damage. It also sort of lubricates the soil environment, making it easier for that root to move through the soil. And as it's shedding all of these cells, it's providing nutrients and exudates out into the soil environment, where the root tip can then interact with microbes, which could include fungi or bacteria or archaea, or even other organisms that are living in the soil and would have some kind of role in that plant's development, whether it be positive or negative. So many roles that the root cap has, but these cells are constantly sloughed off. So they're made as the root cap is dividing, or the root apical meristem is dividing. They get produced, pushed out, and then as the root pushes through the soil, they get kind of peeled off and left behind. So it's an important additive to the soil environment. So that's where the root apical meristem produces cells to the outside. To the inside, these are going to be the cells that produce all of the primary meristems. So we have our apical meristem, and that is going to make primary meristems and a root cap in the root. So these interior cells are going to differentiate into the protoderm, which is one of our primary meristems, the ground meristem, and the procambium. So those three meristems are primary meristems that will make primary tissues. Here we can see a real long section through a root cap, or I'm going to keep calling it a root cap, through a root tip. And here we see the cartoon kind of colored version that highlights what those different regions are. So out here we have the root cap. In here in the center we have the procambium that'll result in vascular tissue. Out here we have the ground meristem. And then the protoderm would be this external layer that is going to make the epidermis. And here we can see those primary meristems developing into the primary tissues, which are the epidermis, ground tissue, which as we know can be divided into cortex and pith, or it can just be ground tissue, and then the vascular tissue, which as we know is xylem and phloem, and then there's other cells that are involved in xylem and phloem tissue, so it's complex. Okay, so here is our root tip long section, including all of these regions of growth. So at the very tip down here, we have the region of cell division, or just the region of division. This is where all of the meristems, so the root apical meristem and the three primary meristems, are just actively producing more cells. But those cells haven't specialized to any function yet. They're still small, freshly made, getting pushed out of the way as new cells are being made below them. So this is an area of active division. As we saw, all of the cells are really small. We can see their nuclei in here, so there is a ton of division going on. Above that, and you can think of this as like a time series, the root tip represents now. Now the root is growing, making new tissues. Those tissues are made now. As we move up this root tip in our diagram here, we're kind of going back in time. These tissues were made earlier in time. And so they are now growing when they're in this region of elongation. And then once we get to the region of maturation, they're specializing into particular cell types. So if you're in the epidermis, you might be specializing in becoming a root hair. If you are in the cortex, you might be specializing in becoming some kind of storage cell. If you are in the vascular tissue, in the xylem, you might be developing into a vessel element, or if you are in the phloem, you might be developing into a companion cell or a sieve tube element. Those are all specialized cells that would start to develop within this region of maturation. The cells would have gotten larger, kind of grown to maybe what their final size might be, or approximating that, within this region of elongation. So, Region of division, massive cell division, making lots of tiny cells, just making the number of cells that we'll need for these mature tissues. In the region of elongation, those cells get bigger, and in the region of maturation, they specialize into whatever their final job is going to be. So think of this as like you're born and you're growing, and then here's your teenage years where you suddenly get really big, and then here's your adult years where you have to figure out what it is you're going to do. So here's a few diagrams of that. Where should I put myself? So these are 3 different looks at a long section. This is a actual long section that's been stained, so you can see the nuclei within the cells. You can see we have tons of tiny cells down here actively dividing. The root cap extends quite far up this growing root tip. Basically where the root cap ends on the sides is where I say the zone of division stops. So the root cap sort of protects that whole zone of actively dividing cells that don't have any other specialized protection. Above that, our cells start to get larger. We see less densely packed nuclei. This would be our region of elongation. But we don't really see a distinct region of maturation here. At least in this image, it's difficult to make out any cells that have specialized. Whereas here in this drawing, we see some of these cells have specialized to become root hairs. You can see these like helical wall thickenings here of the secondary wall of the xylem. We see those vessel elements have also started to specialize. So up here, we're in our region of maturation. This is an even further out view where we have our region of division, where the root cap is, elongation between the root cap and the root hairs, and then this root hairs region is the region of maturation. So that is our root tip that we've been looking at so far. Above that, we can get the production of other roots that are going to move out sideways from this root. So as it gets older, it develops. New root apical meristems can be produced and start growing a new root out to the side, so lateral roots. And notice that these are produced from the interior of this root. So we'll go over all these tissues and where these lateral roots come from, but they have to burst out through the cortex and through the epidermis to make it out of the root. So it's a really interesting way that these lateral roots are formed. So here you can see these lateral roots. We say they're produced endogenously. So endo meaning inside, genus, generate, that to produce something, right? To make something. So they are making these roots on the inside, endogenously. And they are coming from this region of the vascular cylinder. So in the vascular tissue, we get the production of these lateral roots, which sort of makes sense, right? Because you have this major vein of tissue that's sending water up and phloem photosynthates down. You need to connect all of your other roots to that, so you can just grow them starting from that main vein and then push them outward. So these are just new lateral root buds here, and then we see these developing lateral roots with differentiation of their tissues, right? We can distinguish now a root cap, the root apical meristem, this region of division here, and we have a distinct vascular cylinder, cortex, and epidermis forming. This over here is close-up of some root hairs. So you can see they're in this outer layer. The outermost layer of the root is the epidermis. And then we get these long extended cells, still a single cell, but just extending outward, increasing the amount of surface area it has to absorb water. And this is a seedling that has germinated, and we can see that radicle, that first root that forms, is covered in projecting root hairs. They're quite long for being individual cells. Okay, so that's where I'll stop us for this part of roots, and then I'll continue with the vascular cylinder and secondary growth in the next video.